Very good afternoon. Salam sejahtera to all. Thank you for coming to our IMMB Visiting Scientist Talk today. This is a hybrid talk whereby Dr. Jeff Wells is physically with us in the meeting room in the IMMB, Faculty of Medicine, UITM. So again, we will have Dr. Jeff Wells, who actually gave a talk on last Friday on the topic on forensic entomology analysis in the estimation of PMI. And today, Dr. Wells will give his second talk in our faculty. Please allow me to introduce our speaker again, especially for those who are new to this talk. Dr. Jeff Wells is an associate professor in the Department of Biological Science and International Forensic Research Institute, Florida International University in Miami, USA. Dr. Wells received his bachelor from University of Washington, Master of Science in Washington State, and PhD at University of Illinois at Chicago. Today, Dr. Wells has published more than 60 peer-reviewed publications, including book chapters and commentaries. And he has graduated 14 master and PhD graduate students. And currently, she is supervising 2 PhD and 4 master students. For professional experience, Dr. Wells was the president for North American Forensic Entomology Association. He is also the editorial board member of the Journal of Forensic Science International. He has served as expert consultant for more than 50 criminal investigations across the state. So without further ado, let us welcome Dr. Wells to share his talk with us. And the topic of his talk today is molecular biology application in forensic entomology, death investigation, and research. Dr. Wells, the floor is yours.
Thank you for that introduction, Dr. Chen, and thank you all for attending. It's been a wonderful visit so far, and I look forward to what we have to do in the remaining time, and I hope that we can continue with collaborations and academic exchange in the future. Because part of this visit is for introductions between our 2 institutions, I thought I would very briefly talk about my own university. If I can advance the slides. When most people know something about Miami, and if you think about Miami, you think of the beach, but we are actually very far from the beach. Campus is on the western end of the city. This is looking to the east. You can see a little bit of downtown there and the beaches beyond. We are a relatively large university. This campus has between 55 and 60,000 students. And when I think about areas that are of common interest to this institute and to my department, one thing that comes to mind is that we both have an interest in mosquitoes and mosquito-borne diseases, and my department is quite strong in this area. I'll just mention some people. Dr. Lee does a lot of international work, especially in Africa, concerned mostly with malaria and the development of novel drugs to combat malaria. Dr. Dariega works on basic biology and physiology of insects, particularly juvenile hormone biology. And Dr. De Gennaro works on the molecular biology of mosquito olfaction and host seeking. And this obviously is relevant for the development of novel insect repellents. So that's something we have in common. Something that Florida International University is very strong in, and which might be of interest to people in Malaysia, is forensic science, all aspects of forensic science. And a few years ago, this was all combined administratively into what they call the Global Forensic and Justice center. This combines all the academic disciplines on campus that are involved in forensic science. It includes nursing, it includes psychology, it includes the law school, and it includes the natural sciences. And this encompasses the program that has been there for decades, the International Forensic Research Institute, which is the traditional academic mission of the bachelor's, master's, and PhD degrees in forensic science. Investigator-initiated research, so standard college professor research. And we also have some fee-for-service labs where you can submit samples for DNA analysis or toxicology analysis, et cetera, for a fee. But we do a great deal of training, at the National Forensic Science Technology Center. Big facility, trains law enforcement, trains military. A lot of teams come from other countries. This has mostly been Latin America because Miami is a Spanish-speaking city. Probably 2/3 of the citizens there speak Spanish at home. So it's very easy to interact with people from Spanish-speaking countries, but also they do a lot of training with North African countries and the Middle East, and I hope in the future with Asia. They also do a lot of technology evaluation. So a company invents a new instrument, they need an independent performance evaluation. So NFSTC does that. And I don't know if you can read the writing here, but the Center for Advanced Research in Forensic Science, this is a U.S. National Science Foundation sponsored and supported network to promote industry academic partnerships. And it's something like a Shark Tank, if you know that term. Academics get a few minutes to pitch their ideas to industry sponsors, and they give pilot grants in response to this. And if it works out well, then the researcher and the company that can then go off on their own with product development or some new analysis. So now to molecular biology and forensic entomology. I don't have time to talk about all of my own work today, and I'm not going to talk much about the research of other people. There are of course other people in the world who do molecular biology research for forensic entomology. And in our own projects, for the last few years, we've become increasingly involved in generation and annotation of whole genomes, transcriptome, and other kinds of gene expression analysis, including real-time PCR, and just very recently doing metagenomic surveys of the microbes associated with flies. This work is either too boring or too new for me to talk about it yet. Hopefully in the near future, we can visit this again. I understand that most of you are not forensic entomologists, so I'm going to talk very, very briefly. Forensic entomology is many things, but mostly it is analyzing entomological evidence to help in an investigation of suspicious death. And of that, it mostly is about using insect evidence to narrow the window of time when death occurred. And mostly that involves estimating the age of a carrion insect that you believe has been associated with the corpse since that insect was an egg or a newly deposited larva. So age estimation of an immature insect, an egg or a larva or a pupa, is the main task of a forensic entomologist. And you do that by reference to usually laboratory development or growth studies that record development rate as a function of the species involved and the temperature involved, and often other environmental factors. And that connection between development rate and species is very important. Insects grow at different rates depending on their what species they are, and they behave in different ways. So to do the analysis well, the first thing you have to do is analyze this, is to identify this specimen. which is what entomology is very much about. Learning the taxonomy and learning identification is very crucial to most aspects of entomology. And that's a bit of a problem. And this just shows you a typical range in development rate of closely related species. It can vary by about 50% when comparing two members of the same genus. If I had given you some other genus, it would have been about the same in magnitude. So if you misidentified a specimen, you could be quite wrong in your conclusion. Or if you didn't misidentify it, but you couldn't identify it, you would have to be very vague in your conclusions, because you would have to say, well, if it's this species, it would be this answer. If it's this species, it would be another answer. So to do it right, you need to identify the specimen. That is a bit tough, because adult insects, it takes a lot of expertise to identify adult insects, but at least they're anatomically complex, and there are a lot of diagnostic characters. The immature stages have much less to go on, and there is some, I can look at this lower left-hand corner picture, and I can identify the taxonomic family looking at that, and maybe subfamily in the lower right hand. This is the same structures you see here. These are the openings to the respiratory system. Here they are. And I can look at that, and I can tell that this is a blowfly, and I can tell that it's a third instar, and I can tell what subfamily it is, but I can't take it to the species. Then at the other end, there are the mouth parts. So there is some anatomy to work with, particularly with the older instars, but it is often quite difficult. So quite a while ago, decades ago, we worked out how to identify a lot of species based on mitochondrial DNA sequence. And many other branches of zoology are doing the same thing. So there are a lot of technical advantages to analyzing mitochondrial DNA. It's high copy number, it's haploid, and it resists postmortem degradation. So it's easy to analyze, easy to generate sequence data, and it works with very degraded specimens, which is often the case with forensic analysis. And we usually use the protein coding region shown on the left, which is cytochrome C oxidase subunit 1, or CO1. More rarely, the AT-rich region, which is a non-coding region shown at the top. And also, some people use cytochrome B, but that's more appropriate for vertebrates. Now, several years after this became common in forensic entomology and also other areas of zoology, somebody gave it a nice name, called it barcoding, DNA barcoding. And I think most people are somewhat familiar with this, the idea that you do identification or species discovery based on cytochrome oxidase 1 sequence and phylogenetic analysis. So I won't say much more about this, except that it's quite common. But if you're thinking of doing this, because the technology is very easy. It's very easy to do PCR. It's very easy to do sequencing of PCR product. It's very easy to do phylogenetic analysis, but realize that you have to be careful. It works for some groups. It does not work for some other groups. So again, if I talk about, so Chrysomyini is a subfamily of blowflies of Calliphoridae. And I won't go into the details, but just say that extensive validation, analysis of hundreds of specimens involving all of the forensically important species in North America. And there's never been a misidentification. There's never been an identification error when we have run validation studies on this. So it appears to be very robust and trustworthy. Whereas another very prominent group, the green bottle flies, it doesn't work at all. Very curious. And this became apparent to us at first, many years ago, in collaboration with Jamie Stevens and Richard Wall in the UK. And we were analyzing this particular combination of two species, Lucilia sericata and Lucilia cuprina. And these two phylogenies involve a lot of specimens. You don't see the individual specimens, but on the left, you see a phylogeny made with the ribosomal 28S gene, which is a nuclear gene, and on the right is cytochrome oxidase 1, mitochondrial gene. And what you see is, for Lucilia sericata, it is monophyletic for both analysis in both genes. You see one branch leading to all of the specimens of that species, based on that phylogeny. And that's good for the analysis. That works, the analysis. Whereas Lucilia cuprina is what we call paraphyletic. So there's a bunch of individuals here you don't see. And if you were to analyze them, their mitochondrial DNA is more closely related to Lucilia sericata than it is to members of its own species. This is called paraphily, and it is a pattern that makes this unsuitable for using that gene for species determination. Now, when we first did this a long time ago with a limited data set, we thought there was a geographic connection because all of those problematic specimens were from Hawaii. So we thought there was some connection between being Hawaiian specimens and this problem. But additional research showed that this is a worldwide phenomenon, and not just for cuprina and sericata, but for all Lucilia species and closely related green bottle flies, for which we had enough data. And the next slide is going to be a very complicated phylogeny, so bear with me. I just want you to notice that, for example, down here, we have Lucilia porphyrina, and it is more closely related to Lucilia ampullacea, so that's paraphily. Down here, we have Lucilia illustris, and that is more closely related to Lucilia caesar than to other Lucilia illustris, so that's paraphily. And the cuprina sericata, and now Hemipyrelia, that's just a big mess. And so in other words, we found cytochrome oxidase 1 paraphily in every comparison for which we had enough specimens to discover it. So it just doesn't work. And it's looking like some other blowfly subfamilies, it doesn't work so well either. Now, this is not because these are not good species. It's not because Lucilia porphyrina is actually Lucilia ampullacea. These are good species in terms of morphological analysis. And if you do a more extensive DNA analysis, it comes out okay. So remember, let's look at Lucilia illustris and Lucilia caesar. We followed up on this with AFLP analysis, which I will describe later in the talk. But these are complex nuclear genotypes, and Lucilia caesar and Lucilia illustris are monophyletic with this analysis. So this is now routine. Using DNA to identify specimens is reliable if you've chosen the right group. And if you can do mitochondrial DNA analysis, you should, because it works really well. There are lots of protocols that you can use, and you get nice, clear data, as long as it is a taxon for which it is useful. Something else that was developed quite a while ago is used much less often, but is used sometimes in death investigation, is to analyze vertebrate DNA, including human DNA, that you get from the insects. And one source of insects is the gut of a maggot. This is the first way we demonstrated it. Now, later on, I'll talk about analyzing DNA from adult insects. So why would you want to do this? Why would you want to analyze a maggot's last meal? There are a variety of reasons. Probably most often is when you discover a scene with maggots, but no corpse. So someone reports a bad odor, and police go in, and there's no dead body, but there isn't. There are a bunch of maggots. Those maggots were feeding on something. You would like to know what it was, and if it was a human, you would like to know who it was. Another situation that arises relatively often is investigators find a corpse. They identify a suspect. They go to the suspect's house or search the suspect's car, and they find maggots, suggesting that that might have been the murder scene, and then the corpse was then transported. So if you can analyze the stomach contents, it's not really stomach, the gut contents of that maggot, and connect it to the corpse, that's very incriminating. That places the corpse at the scene, and it would look very bad for the suspect under those circumstances. Also, remember, if we're going to use maggots for estimating time since death, you must be sure that the maggot was associated with that corpse all of its life. But that's not always obvious. So sometimes you find maggots in a corpse, but they're not in direct contact, making you absolutely, you're not sure that they're from the corpse, or they're in contact with, there's another potential food source nearby. There's another body, there's a dead dog, there's a dead rat, something like that. Maggots can't crawl very far, but they can crawl. So it is possible, if there's a dead dog nearby, that the maggot crawled from that dog to the human corpse. If that happened, then you couldn't use that specimen for estimating postmortem interval. And so we have shown that when that happens, if we did this artificially, transferring maggots from one food source to another, that you can still get the signal of that original food source for a couple of days. And so we looked into this, and this is something that it didn't occur to me at the time we first published this. Sometimes you get what we call a chain of evidence dispute. In other words, someone will claim that the maggot specimens used by the police, those are not the maggots that were collected from the body. You know, they'll say there was some record keeping problem. And then you can use DNA from those maggots to connect to the victim and say, yes, these are the larvae that were taken from that victim. And so we do this by taking advantage of a handy part of gut anatomy. Maggots have a crop. So this is the mouth right there. This is the dissected alimentary canal. You have a crop, which is a blind sac, which fills up with a relatively undigested tissue. This picture is a bit bleached out, but you see these maggots on a knife wound. You see that red spot, that's a crop. You see on this picture, that's the crop right there. So it can be relatively large. It can be, 1/3 or so the length of the body. So if preservation is good, you can dissect this. We do maggot surgery. And then from then on, it's just standard forensic DNA protocols. And down below, this just shows that we're able to amplify the HV1 is a human mitochondrial DNA locus, and CO1 is the maggot mitochondrial DNA locus. And you have no problem because the primers are very different. You can amplify them both separately. And just to show you simple example, we had a fellow who had a liver transplant, and because he was on the medical school staff, he was able to keep his old liver, so he gave it to us. We fed maggots on that liver. We got a blood sample from him. We genotyped both. And this is just mitochondrial DNA sequence data. We did the same thing with short tandem repeat loci, which is standard forensic DNA analysis, and they match. And it's no surprise, but it's good to demonstrate that. So, and this is done now. It's not done very often, but this is occasionally a part of criminal investigation in my country. Now, more recently, people outside of forensic science have been using carrion insects for DNA analysis, for vertebrate DNA analysis from the adult in a very interesting way. And this is because you can use carrion insects as biosensors. You can use them to sample environmental DNA to survey, for example, for vertebrate animals that are present in the environment. The flies do a better job of finding rare or shy vertebrates than humans do. And then you can catch them. And this is a big advantage of carrion insects, is that you can trap them. You can attract them to a decayed meat bait. They can fly very far in one day. We don't know how they do on average, but they can easily fly kilometers per day. And I'm just going to show you a little video of trapping them. This is me in my backyard. And it's not going to happen, is it? Okay. So first I open a decayed meat bait and put it in the bucket, and then just place the net over top. And if you do it that way, then the insects coming in will never touch the bait and so not be contaminated. And I'm afraid it's going to be about 30 seconds of nothing happening. But you watch carefully, and I get in about 30 seconds from when I open the bait, you'll see the first insect make a pass. Wait for it. there, the first one came by, and then they're starting to go on top of the net, and then they start to land. And then it's relatively easy to catch them. Once they get past the edge of the net, you just quickly swing the net upwards and you can catch them. And so that's very easy. So you could go to an area that is very hard, very inaccessible, very thick vegetation, and put out a bait, and you'll attract flies. And those flies will have been out in the environment, and they land on carrion and they feed upon it. They land on feces and they feed upon it. And then you can analyze that material. And it's found that this is better for surveying vertebrate animals than trapping or direct observation or motion detection and motion activated cameras. And I'm going to go through just a bunch of papers here just to show you that quite a bit has been published on this, and it's becoming very useful with this application. So I mentioned earlier this idea of using gut content analysis to reconstruct the movement of a corpse after death. And this is not an unusual thing. Often the place where the corpse is found is not where death occurred. But the DNA within a maggot gut, depending upon the circumstances, may only last a day or so. And so we wondered if there might be another way to approach this if there was no vertebrate DNA available. And so we thought we would do this from a population genetics standpoint. And this has to do with the natural history of blowflies. So a female fly, once she has matured eggs, so she might have 150 to 250 eggs in her abdomen. She finds the corpse through olfaction, through the odor, and lands on it. And if she's undisturbed, she'll lay all of those eggs within just a few minutes, maybe 10 minutes or so. And then once she's done, it will require at least another week for her to develop new eggs. As a result, if you were to find maggots at two locations, so remember the example of you have a body found out in the brush, and they have a suspect, and they go to the suspect's house and they find some other maggots, and they are the same species and about the same degree of development, so about the same size. If you were able to demonstrate that those are full siblings, there's only one way that could happen: if the same corpse had been at both locations. Okay? So if we could do kinship analysis, essentially what we're proposing here is maggot paternity testing. And so to do this, we needed to, this is a population genetic analysis. In humans, where paternity testing is and kinship testing is quite common, we usually use microsatellite loci, also called short tandem repeats, or STRs. So this would be a standard way to do this. This is just to demonstrate it. You have a body with maggots on it, the body is moved, maggots are left behind, and then later you find a corpse with maggots. So if you can show that this maggot is a full sibling of that maggot, then there's really no other way that could happen unless the corpse was at the same corpse was at both locations. So when we started, we couldn't do STRs, which is the, as I said, the standard way to do this in humans and in a lot of other animals. So we adopted an analogous technique. So this is called amplified fragment length polymorphism genotyping. It is a method that can be applied to any genome. You don't need to have any reference information about the genome, unlike STRs, in which you have to have the protocols all worked out. And it starts as do many molecular biology procedures with restriction digestion. We extract the DNA, digest the genomic DNA with a combination of two different enzymes. And these create sticky ends, if you know what that means, to which we ligate short two-stranded oligonucleotides. And now we have installed primer annealing sites, PCR primer annealing sites on the end of these fragments, which you can use to amplify the fragments. You don't want to amplify all the fragments. If you did that, it would be too many to analyze. So we go through a process of reducing the number of these fragments that will eventually end up in the analysis. And you do this through what we call selective PCR. And selective PCR means, so this would be the primer annealing site. You know that sequence because that's artificial DNA that you made or bought. If the PCR primer overlaps past the ligation to the linker site and into the original DNA, then it will only match in some of your fragments. So it will only amplify some of your fragments, those in which you have base pairing matched there. And so you do this a couple of times. You do pre-selective PCR, then you do selective PCR where the primer may overlap one base into the flanking region, and therefore it only match 1/4 of the fragments. And then you do it again where it goes 2 bases into the flanking region, then it will only match 1/8 of your fragments. And you do this however many times you want. And then in your last PCR, you use a fluorescently labeled primer so that you can then measure fragment length using capillary electrophoresis. And with the same DNA extract and same restriction digest and adapter ligation, you can use different primer combinations and generate a different profile. And you can very easily generate quite complex genotypes. You can easily have like 600 alleles. We did more like 250 with any organism you want. And these are anonymous. So this peak right here, it's an allele, it's present. We have no idea where that is in the genome. Okay? We just know that it's a certain number of base pairs long and it exists. And this is very robust. You analyze the same DNA twice, you get the same profile, almost 95% similar. So one thing that people don't like about this is they don't like the fact that these alleles are anonymous. You don't know where in the genome they are. Also, you might think that this tall peak is homozygous and this short peak is heterozygous, and that may be true, but you can't tell for sure. So you cannot, at least the last time I checked the literature, distinguish a heterozygous from homozygous dominant. You can tell if it's homozygous allele absent, but if the allele is present, you don't know if that is a heterozygote or a homozygote. So you cannot get Mendelian genotypes out of this, and you can't do a lot of the standard analysis, like F statistics or something like that. But that's not really a practical problem. Again, some people don't like AFLP. They'd rather do microsatellites if they can, because they would like to know the exact genotype and do familiar statistics, but it's not a practical problem. And again, that's why they don't like it. It's not a practical problem because you can generate such complex genotypes. Again, hundreds of alleles. You can do anything you want. You can do with gene flow studies. You can do kinship analysis. You can look at inbreeding. Anything you can do with other loci, you can do with AFOP, and there are no practical differences. Now, of course, you have to have very high quality DNA. So this is something that wouldn't work with a lot of forensic samples with a degraded sample. It's more of a research tool than an investigative tool. So we started doing blowfly population genetic surveys, and we have done this with Formia regina, with Lucilia cercata, and with Chrysomya megacephala, and the results are all quite similar. And so we'd go out and attract flies to a bait in the manner that I showed you before, store them in ethanol. We do the digestion with these two restriction enzymes, and it would generate about a 230 locus or allele genotype. We'll talk about the population genetic analyses later, but one thing you get is allele frequencies, which is a basic population genetic parameter and something you can do a lot with. For example, you can also work on paternity testing if you know allele frequencies. So this would be for the wild population, and what you would expect to see with non-related individuals. If they're from different locations, you have no reason to think that they're at least closely related. So you know what the expected amount of shared alleles between non-related individuals. What would you expect to see with full siblings? So we just produced full siblings. We took individuals from very different locations and crossed them in the lab and produced full siblings. And then we used an index of relatedness, R. R is a function of the proportion of shared alleles. And it, like a lot of indices, it goes from -1 to +1. -1 if there are no shared alleles, +1 if they're completely the same genotype, and 0 if they have the proportion of shared alleles you would expect given the allele frequencies in your population. And with that, we were able to generate frequency distributions for R, for the wild population of unrelated individuals, and for full siblings. And we got this. Shown on the left are the frequency distributions of relatedness for unrelated individuals, and very nicely it centers on 0, which you expect it to be. And these are full siblings, and nicely it centers on about 0.5, which is where you would expect it to be. And with these data, you can make a likelihood ratio test. You can calculate the likelihood of observing a particular R value. Remember, R value is a pairwise comparison. So you have two individuals, you calculate R for the comparison of those two individuals, and you can do that, you get an R value, and then you can calculate the likelihood of observing that R value if they're unrelated versus the likelihood of observing that R value if they're full siblings. And at about R 4.1, you've reached a situation where it's about you're about 1,000 more likely to observe that value of R if they are full siblings than if they are unrelated. This is how we use likelihoods in forensic science. And we chose 1,000, likelihood ratio of 1,000, because in American paternity testing, that is the more stringent standard that exists. So we can do this. Have we done it? No, we haven't been able to take it further. We haven't gotten any grant funding for this. I don't know if anybody has ever done it in casework, but it's possible, and I hope to get back to it someday. So while we're doing these population genetic surveys, we might as well investigate some other hypotheses. Is there geographic structure? Is there a population genetic structure? That is, are there regional populations? Is there any barrier to gene flow? Is there any regional isolation of these forensically important insects? This is of practical interest because if there are regional populations, this would suggest that they might differ in basic biological properties such as growth rate, meaning you wouldn't want to take biological data from one population and apply it to a death investigation of the other population. So you might need to do additional research. This is not very likely, but if there are regional genotypes, then if someone were to transport a corpse long distance and you were to genotype a maggot from that corpse and say, this is not a local genotype, this is from someplace else, you would infer that the corpse had been moved. I don't know if that's ever likely to happen, but it's theoretically possible. So looking at these surveys, and again, we've done this for 3 forensically useful species, and these are just standard population genetic analyses, molecular analysis of variance, Mantell, so this look for regional population structure, isolation by distance. So is there any connection between the geographic distance between 2 individuals and the genetic distance? Are they less likely to share alleles if they're far apart than if they're close together? And again, we looked at the pattern of relatedness that I described before. And I have to admit, when we first started doing this, I didn't think it'd be very exciting. My student really wanted to do it. It turned out she was right. We found out some kind of interesting things. And so this just shows the Formia survey, samples across the continental or what we call the lower 48 states in the USA. We arbitrarily divided this into 3 regions, somewhat connected to the topography of the land, and then did the analysis. So what did we find? There was no geographic structure. There was no difference between flies collected in California versus flies collected in Virginia. Just no regional populations at all. But strangely to us, the samples themselves were distinct. By sample, I mean the flies you collect at one bait over 30 minutes. Those were all distinct. Every single one was distinct. Every single one from all 3 species was distinct in that those individuals shared a higher than expected proportion of alleles. They were related. All of the samples appeared to be closely related. There was no exception to this. It's like a biological law. So what is going on? I didn't expect this at all, at least at first. So what would cause this? And by the way, you could collect in one place at different times. You could collect at one spot for 30 minutes, and those individuals would share a non-randomly high proportion of alleles. You come back 2 weeks later, collect again at the same spot, and those individuals would share a non-randomly high proportion of alleles, but they wouldn't be at all similar to each other. They would be as different as if you had collected flies in California and in Florida. So what is going on? Well, one possibility is that flies, blowflies, move in family groups. We don't know this for sure, but that would explain the pattern we see. And this seemed crazy to me at first, but in retrospect, lots of animals move in family groups. Of course, lots of animals start life close to their siblings, and they stay close to their siblings. So what could happen there? This is pure speculation, what I'm going to talk about here. So remember, a female fly deposits about 200 eggs at once. They'll feed in the same corpse or carcass. They'll pupate in the same place, and they'll emerge as adults at about the same time in the same place. They are teneral, that is, they're not completely finished with metamorphosis, and they can't operate very well, so they'll be very shy and hide under leaves. And then they'll become ready to fly around, in which they want to seek carrion at the same time. Carrion is relatively rare in nature. There are not that many odor plumes. So they would probably run into the same odor plume, track it to the same carcass. So the fact that their first carcass visit would be by a bunch of full siblings is, in retrospect, not so crazy. But we found that this even works for females who are gravid, that have a full of mature eggs. And those females have visited at least 2 carcasses, because they visited the first carcass and they got a protein meal. And then later, once their eggs were mature, they found a second carcass. And the same pattern exists for those. So if this is true, this persists through multiple carcass visits. And it's interesting and it's unexpected. So finally, I'll talk about our current research. And I have to admit, although I made my career based on molecular biological applications for forensic entomology, I became quite bored with it. I didn't care about it anymore. I wanted to work on statistics. Of course, the students needed to learn this, and so I kept attracting students who were interested in learning genotyping techniques for good reason. The students need to learn to do this because it's what they need to do to get jobs. But recently, I've become excited again based on molecular genetic engineering of carrion flies. And of course, this is highly regulated research. We have a biosafety committee at the university that watches everything we do, but I still get a lot of people who are afraid of this. But I would just mention, it's too late. I ask you, do any of you here keep aquarium? Do you have a home aquarium? Do they sell glowing fish here in Malaysia? Do they? Okay, yeah. So these are aquarium fish that will glow under blue light. This is the same technique that I'm talking about for life. These fish have been genetically engineered to express a fluorescent protein under blue light, and it's already out there. So I just wouldn't worry about the flies so much. So why do we do this? The problem is that empirical validation of maggot age estimation under realistic conditions is difficult to do. So what we have is growth data in the lab. But those conditions are very different from the field, where you have huge populations of maggots. It's a mixed age, mixed species situation. It's humans. Is the growth rate the same? We're not sure that it is. So we would like to perform validation of maggot age estimation in a crime scene conditions. But if you just throw out an animal carcass or a human, which we can do, and you look at a maggot from that body, all you can know is that it's not older than the time since exposure. You can't know its true age. How could you know its true age? Well, if you can mark a maggot, if I could take I can get eggs from a colony, I know exactly how old they are, and I can put them on the corpse. If I can mark them in a way that I could find them again later, I would know their age because I know when the eggs were laid. But you need to mark them in a way that will persist despite the fact that they grow tremendously and molt repeatedly. And because I'm running out of time, I'll just get right to the point, which is we can mark them by insertion of a gene for a fluorescent protein, which we do using Formica regina, the most common forensic evidence in North America. And we do this. We tried CRISPR, if you're familiar with that. We tried to do a CRISPR knock-in, but we just killed our flies. And then we tried a variety of transposable elements. And finally, somebody gave us some that would work. And this is a piggyback transposition system, which uses two DNA plasmids. One has the transgene, which is for the fluorescent protein, and the other is for the transposase, which causes the insertion of the gene into the genome. This is random. You don't know where it's going to go, and it could go in more than one place, and it could go in a place that causes lethality, or it could go in a place that doesn't cause any problem at all. If you're not familiar with insect embryos, they're very yolk rich, and so what you get is early in embryogenesis, you get the division of nuclei without the formation of cell membranes, and then they move all the way to the periphery, and then they form cell membranes. And this is where the germ nuclei are. And of course, we want to get insert the gene into the germ nuclei so it'll be passed on to the offspring. So this is the target, the posterior pole, prior to the formation of cell membranes. And on this diagram, which must be Drosophila, it says 3 hours. For us, it's more like 90 minutes. Blowfly eggs are deposited in a clump, cemented together. And so this is a very simplified version, but we get eggs from the colony in just a 10-minute interval. They're separated and decoriated. That means take the shell off using NOH, I should say bleach as well. I don't know why I didn't put that on there. And then they're desiccated because you're going to be injecting fluid into the embryo. And if you don't desiccate them, you'll destroy the egg with the pressure, and then micro-injected with the donor and helper plasmids. And it turned out we had to do this by about age 1 hour. This shows you the glass needle going into the edge of the egg. And then afterwards, we screen them in the G0 generation for fluorescence using a fluorescent microscope, UV light and a red filter. And those that show positive then have to be back crossed with wild type for a few generations in order to get individuals that have a insertion in only one place, and also you get rid of some deleterious effects that way. And they do this for a few generations, and then we in-cross to produce homozygous individuals. And this was terrible. We had failure for a year. The most we would get would be some mosaic expression, which means it's somatic. It's not in the germ line. Also, this is during COVID, so research is difficult in general. And we were getting very depressed, and we were getting ready to give up. when a single injection succeeded. Okay, so we don't even know why it succeeded because there's only one, and we changed methods during this time, and that might have been responsible. So it might not be 1 out of 4,000. It might be 1 out of, say, 1,500 with the new method. But we don't know. But this is right towards the end. We were ready to give up. A single injection produced. You see, this is a white light, the wild type individual on the left, transgenic on the right. Look at under UV light with a red filter, and they glow like crazy. They're beautiful. This shows you at the top, again, under white light with wild type on the bottom, and on the right, you see the homozygous individual at the top, heterozygous at the middle, so you can see the difference. And this is not something we cared about, but it's kind of interesting. You can see inside the pupae. You can see the developing fly inside. So this thing's still alive. You can observe metamorphosis based on that. So now we finally got it, and we're not going to let these things go. And the next step will be field experiments, which is going to require a lot of careful planning because we obviously cannot release transgenic individuals to the wild. We have to make sure they don't escape, but we have to keep things as natural as possible. So there's a lot of work in the future in order to put this to use, but we now have what we need. And by the way, you can see the fluorescence just with a cheap blue flashlight and wearing orange sunglasses. You don't need a fancy equipment to do this. So with that, I've gone a little bit long. I'd like to thank all my colleagues. We have a blowfly genomics team with 2 professors, 2 research faculty, 2 PhD students, especially Amber McInnes, who this is her PhD dissertation. So she has done almost all of this work, but Mason Lin has been very helpful because he does the supporting genomics and transcriptomics work, which helps us support this. The population genetic work I described was done by Christine Picard, now at Indiana University, Purdue University, Indianapolis, when she was my PhD student at West Virginia. And we had tremendous help from Dr. Max Scott from North Carolina State University, who developed a lot of these protocols and provided us with those piggyback elements. And of course, we are eternally grateful to our funding agency, and I'm grateful to all of you for listening. Thank you very much.
Thank you very much, Dr. Wells, for your very interesting talk. Now we are in Q&A session. For participants online, you can write down your question in the chat box, or you can ask questions directly to the speaker by switching on your microphone. Yeah.
I like your main bar chart.
That open the bucket. You actually jogged something in my memory because that also prevents the flies from touching the bait, which means you won't introduce new bacteria. And because of that, I realized that I caught my flies wrong. So now I'm going to go back and check them because that will prevent the flies from being introduced to new bacteria. So I want to thank you for actually making that homemade trap. That was extremely helpful.
Yeah, that's our, well, our original purpose was just not to get the net stinky. But since then, it works for the same way to prevent DNA contamination or microbe contamination.
Yeah, extremely helpful. So I'm going to go back and retest using that method.
Okay.
So thank you.
And you want to somehow sterilize your net in
Yeah.
between.
Yeah.
Okay.
Thank you.
Well, I'm glad that worked. People listening was that the collection technique I mentioned prevents contamination of the incoming flies by the bait, which can be useful for your purposes.
Yeah. Any more questions? Yeah, please.
Thank you for the informative talk. I'm not a forensic entomologist, so I know very little about it. I'm really interested in age estimation. So, is there any parameters that you look at either insects or age estimation?
Yes, so the question is, how do you the age of an insect?
Yeah, adult insects using morphology.
Oh, adult insects.
Because
So that's much more difficult. So there are some changes in the cuticle. And the most common way is not really age, but ovarian cycle. So a lot of people who are interested in blood feeding insects are interested in how many times they've fed, because that's important for disease transmission. And there are characteristics of I can't remember now what they are, whether you can tell how many blood meals they've had, or whether you can tell how many ovarians, probably how many ovarian cycles you can go through. So they don't know the age, but they know this individual is fed on 3 separate times. But age estimation, except for very early in adult life, because the insect's going through a lot of changes still. It looks like an adult insect, but at that point, it's actually half larva, half adult. And so for 5 days or a week, it's still going through a lot of changes. So you could easily, I think, age it during that time. And there are like layers added to the cuticle and some. But I'm being vague about this because we are mostly concerned about immature insects. And most of our attention is at how to tell the age of an egg, larva, or pupa. Okay.
Is there possible reason that you're looking at young insects as compared to adult insects? Because aren't they all on the carpets?
Right, but in the most common analysis, the adult is not necessarily associated with any particular time-sense death.
Okay.
So whereas the larva, which was deposited on the corpse as an egg and stays with the corpse all its life, adult insect can come and go. It could be there on day 1, it could be there on day 5, whereas that maggot has been on the corpse all its life. So if the maggot's 3 days old, and almost always the person was dead when that started. So you can say, well, dead for at least 3 days. Okay. I see something in the chat.
Oh yeah, there's one question in the chat box from Abdul Rahman Kazim. Hello, Dr. Wells. Thanks for the presentation. I have a question. Is AFLP recommended to investigate the issue of the species complex with various taxa?
So we found it useful. And it only works with very closely related individuals because you couldn't do a phylogeny of all the species in a family because not very closely related individuals wouldn't share those loci. So it's not like a conserved sequence where you can align the sequences even though they're not very closely related. You can determine homologous characters. So it doesn't work for that. But if they're very closely related, then yes, it's been done, for example, in blowflies with protocolifera, the bird blowflies, which are taxonomically very difficult. AFLP has been applied to sort out the phylogenetic relationship to that. We've done it with some Chrysomya that it had been misclassified using mitochondrial DNA. And in the example I gave today, we applied them to the sister species, Lucilia caesar and Lucilia illustris. So if you're dealing with very closely related species, yes. It won't work if you're dealing with different genera.
Okay, thank you, Dr. Wells, for your answer. Is there any more questions from our participants? There's one question from Zulaika Roslan. Very interesting topic. As a biochemistry PhD student, I'm not familiar with forensic entomology. However, CSI is my favorite series. So I'm wondering whether all of the technologies in the forensic series on TV were real.
No. It's not ridiculous. What's particularly unrealistic is that it's in CSI, it's just so fast. You know, they put something in a tube, they put it in the machine, it spits out the answer right away. But if I may give my own opinion, I'm not bothered by the science. But what bothers me is that in those kind of shows, the forensic scientists are scientists and they are cops. And that does not happen. If you become a forensic scientist, you will not interrogate a suspect. And so that's what I think misleads. We get a lot of students who go to school and want to get a forensic science degree, and they have an idea they're going to become like somebody on CSI. And the big disillusionment here is that they're not going to become forensic scientists and cops. You know, so that's what I think is the worst disinformation you get from these. You might go to the scene, you might collect evidence at the scene, you will do the analysis, you'll go to court, so you'll feel the satisfaction of following a case through, but you'll never arrest somebody and throw them in jail. And that just does not happen.
Okay, thank you, Dr. Wells. Yeah, there's no silly question actually. Yeah, so yeah, we encourage more questions from participants. All right. Due to time, now it's already 1:01. I think we can conclude this session, Dr. Wells. If you have any more questions to ask Dr. Wells. There's one more from Dr. Sean Lee. Is it okay, Dr. Wells, to answer this?
Oh, certainly.
Yeah, so just wondering if the piggyback proteins was detected in the mole or just in the larvae. Also, was it eventually observed in the adult fly?
So if you mean the cast skin, no. I didn't see it in the cast skin. Yes, it is in the adult fly, although the adult fly is highly pigmented, so it's not so easy to see. So you can only see it in the inner segmental membranes, and of course you can't see it in the head because the eyes are highly pigmented. So yes, you can see the pigmentation in all life stages. The eggs light up like Christmas lights. Interestingly, also, the eggs are fluorescent even if they don't have the genotype, as long as, so say the mother is heterozygous and or the mother is homozygous, you cross her with a wild type male, so only half of her offspring have the transgene, but all of her eggs fluoresce, meaning that she is passing either messenger RNA for the gene or the protein itself into the eggs, because they are all fluorescent.
Okay, thank you very much, Dr. Wells, for your answer. On behalf of the IMMB, Faculty of Medicine, we would like to thank Dr. Wells for sharing your research with us. So with the end of this seminar, we concluded the seminar series by our invited speaker. Dr. Wells' talk will be recorded, and you all can always watch it again on IMMB YouTube channel. So with that said, I'd like to thank all of you who are with us today. Please fill up the attendance form. Thank you everyone. Take care, and I see you again. Thank you. Bye-bye.